In this second video that we have on cells, we're going to cover passive and active transport, hypertonic, hypotonic, and isotonic solutions, endocytosis, gap junctions, and apoptosis. Active transport moves ions from a low to high concentration, uses a sodium-potassium pump, and requires ATP. Passive transport moves ions from a high to a low concentration and does not require ATP. Both active and passive transport use ion channels to move ions across the cell membrane and enter into or out of a cell. The sodium-potassium pump functions to pump sodium ions out of the cell and potassium ions into the cell. The sodium-potassium pump is a well-understood example of active transport. Sodium and potassium ions are pumped in opposite directions across the membrane, building up a chemical and electrical gradient for each. These gradients can be used to drive other transport processes. In a hypertonic solution, cells shrink due to water flowing from a higher water potential to a lower water potential. Cell shrinking is referred to as plasmolysis. In a hypotonic solution, cells either swell or burst. In an isotonic solution, they do neither. In an isotonic solution, the concentration of the solutions inside and outside the cell are equal. Endocytosis is a form of active transport in which a cell transports molecules, such as proteins, into the cell by engulfing them in an energy-using process. Endocytosis and its counterpart, exocytosis, are used by all cells because most chemical substances important to them are large polar molecules that cannot pass through the plasma or cell membrane by passive means. Endocytosis includes pinocytosis, which is cell drinking, and phagocytosis, which is cell eating. So we'll go over those briefly here. Pinocytosis, again, is sometimes referred to as cell drinking. Pinocytosis is a mode of endocytosis in which small particles are brought out to the mitochondria and then suspended within a small vesicle. These vesicles subsequently fuse with lysosomes to hydrolyze, break down the particles. This process requires energy in the form of ATP. Phagocytosis refers more to cell eating. Phagocytosis is the process by which certain living cells, called phagocytes, ingest or engulf other cells or particles. The phagocyte may be a free-living, one-celled organism, such as an amoeba, or one of the body cells, such as a white blood cell. In some forms of animal life, such as amoebas and sponges, phagocytosis is a means of feeding. In higher animals, phagocytosis is chiefly a defensive reaction against infection and invasion of the body by foreign substances, antigens. Gap junctions are specialized intercellular connections between cells in most animals. Gap junctions are an example of cell-to-cell communication. They directly connect the cytoplasm of two cells, which allows various molecules, ions, and electrical impulses to directly pass through a regulated gate between cells. Gap junctions occur in virtually all tissues of the body, with the exception of adult fully developed skeletal muscle and mobile cell types such as sperm. Gap junctions, however, are not found in simpler organisms such as sponges. Apoptosis is a process of programmed cell death that occurs in multicellular organisms. Biochemical events lead to characteristic cell changes and death. Triggering signals can come from the nucleus, the endoplasmic reticulum, from outside the cell, and the mitochondria can initiate apoptosis by sending a relay protein to the cytoplasm. It is estimated that between 50 and 70 billion cells die each day due to apoptosis in the average human adult. In contrast to necrosis, which is a form of traumatic cell death that results from acute cellular injury, apoptosis is a highly regulated and controlled process that leads to advantages during an organism's life cycle.